Thank you.
Good. Thank you very much. Professor Mimica has already said the title, so I won't repeat, but it is, now let me make sure, just, why are we talking about this at this time in particular? When I was in Croatia, there were sites participating in our clinical trial, and I'll share with you some of the results from that phase 3 trial. But we are within 8 months of getting the results of the ongoing phase 3 trial. And I'll show you what results we hope to get, and then discuss what implications this would have for treatment going forward. So the company was founded on the basis of 2 key discoveries: the identification of tau protein, in particular, this short fragment here. So here is a full tau molecule, and it's illustrated here in a hairpin conformation, because it's now known that this core unit undergoes a conformational change. And in this state, it forms oligomers, filaments, and tangles. The second important discovery that is the basis of the company is a class of molecules, of which hydromethylthionine is one, which has 2 actions. It disaggregates the existing oligomers because it changes the conformation of the units and releases them. The whole structure becomes unstable, and it blocks further capture of tau protein. So why is tau important? Tau pathology is the most closely linked to cognitive decline. Over the years, there was strong opinion against this view. Amyloid was the way to go, and what causes Alzheimer's disease in many experts' opinions. But when we discovered the tau protein as a constituent of tangles, we were able to show that this is way back in 2000, that there's a close link between the quantity of tau pathology as shown by what is called Braak staging. And I'll explain that in a moment, but it's a measure of the quantity and spread of tau aggregation pathology and decline on mental functioning. This came from a prospective epidemiological study. This cognitive data came from patients in primary care in the last 12 to 18 months prior to death. So Braak staging, in those days, we can only measure it post-mortem, but nowadays it can be measured with tau PET ligands. And as I say, the ligand binding shows the increasing spread and quantity of the tau aggregation pathology. A second marker of tau aggregation pathology is the loss of brain function. So what's highlighted here are the areas of the brain that are no longer functioning, areas of the brain that have gone to sleep because of the tau pathology, have impaired function. And that too follows Braak staging. How many people are affected? These are calculations for 2030, so they're projections based on the European data, Eastern and Western European population data from the Population Division of the United Nations. And you can apply data that was published really from Braak's laboratory and Ohm et al., a colleague of his. And from this, we can calculate what the age distribution is by Braak stage. So Braak stage 1 peaks in Europe at about late 50s, Braak stage 2, mid-60s, Braak stage 3, later 70 to 80, and Braak stage 4 typically for 4 or beyond. So this is all stages 4 to 6 later in life. You can see that if you have Braak stage 1, this is cross-sectional analysis. So, but even so, we can conclude that if you are at Braak stage 1, you may escape progression with a chance of about 50%. However, if you are at Braak stage 2, you will just progress because the total number here at 2 and 3 is the same. And here, the number is less because by that time, about half the population is dead. So to summarize, 48% of the over 45 population have some brain tau pathology, and 26% are at Braak stage 2 or beyond. So just to remind you what Braak stage 2 means, Braak stage 2 is this. It's the point at which people it becomes visible to us as clinicians and to the family that the patient is no longer functioning as they used to. So if we zoom out and look at the whole world, you can see that there are over 500 million, so 600 million people at Braak stage 2 or beyond. These are figures for 2025. And interestingly, only 12% of these are in the US and Europe combined. 59% are actually in Asia. So just a few words about what tau does and how it goes wrong. So the normal, the day job, if I may say that, of tau protein is to stabilize microtubules. So microtubules are made of alpha, beta dimers, and tau, by binding to this dimers, stabilizes the whole assembly. So here it shows in greater detail. Here's the repeat domain. Here's the N terminus and the C terminus. Now, microtubules are critical because they are not only are the structural backbone of all axons throughout the brain, but they also function as a rapid transit system for transporting synaptic vesicles, mitochondria, and other synaptic constituents through to the nerve terminal. So what happens when tau goes wrong? Our theory and others as well take the view that it actually is a product of waste products that accumulate in long-lived neurons. The pyramidal cells, they don't turn over during life, so they live for 70, 80, 90 years, the same single neuron. And it builds up waste products, in particular lipofuscin. Lipofuscin is what's called an age pigment. And we showed some time ago that this, and we have also recent data, that it triggers tau aggregation. So in other words, tau can bind to this and undergo this primary conformational change. And from then, it becomes self-propagating. So we showed that in 96, that it is an autocatalytic self-propagating process. Now, why is that really damaging to synapses? Well, because I told you about the microtubules, but microtubules move slowly down the axon, and they have to stop. They have to come apart at the nerve terminal. So tau is released here. So if there are seeds, oligomers, then more and more will form. So this will gradually destroy the synapse. What's worse is that these oligomers are able to jump from neuron to neuron. There's an important recent paper that shows that if this year, that shows that if there is no synaptic oligomeric tau, there is no dementia. Very important paper. So here I've shown you again the tau protein. what happens to it is that the N and C terminus get truncated. The bulk of the core structure doesn't have the N terminus. So the therapeutic approaches that have been based on phosphorylated tau, targeting that, in our research, phosphorylation of tau actually inhibits aggregation. It's protective. So targeting the phosphodomains of tau is targeting a minority of the tau protein. Over 90% of the tau protein in a tangle is this truncated core unit. And how does hydromethylthionine, LMTM, work? Well, here is the molecule. It's a diaminophenothiazine. And we showed a long time ago that it disaggregates. And recently we've shown that it blocks the oligomers. Following disaggregation, these oligomers are able to be processed via the proteasomal clearance pathway. So the cell can clear the aggregates. Good, so that's all the theory. How does this turn into a practical treatment? Well, it's taken a long time. We did a large phase 2 trial between 2004, 2008, with an oxidized precursor of the hydromethylthionine molecule. And with this, we found that the minimum effective dose, it was a dose finding study. We found that the minimum effective dose was 138 milligrams a day. And we showed in the phase 2 paper that it confirmed that there was significant reduction in cognitive decline and a reduction in elimination of the reduction in brain metabolism that I showed you earlier. So based on that, we designed 2 large phase 3 trials in almost 1,900 patients. And we assumed that doses around here, in fact, we tested 150 to 250 milligrams a day, were the doses needed for pharmacological activity. We thought, and I'll show you this in a moment, that a low dose of 8 milligram would be ineffective and it could be used as a masking agent. So as I'll show you, we found that actually a much lower dose is needed. And that's what the current phase 3 trial aims to confirm. So here we are, and we'll be getting top-line data mid next year. So what did the data from this large cohort of patients show? Well, it showed 2 clear results. And this was published in Shelter et al. 2019. These are the doses that we tested in the phase 3 program. We were testing 150 to 250 milligrams a day, which we thought was what was needed for efficacy. And 8 milligrams a day was used simply to blind, to discolor urine sufficiently so that patients wouldn't know if they were on active. The drug causes some discoloration of urine, a sort of a greeny blue, and the regulators insisted that we have some masking treatment. So we in fact did 2 phase 2 studies to identify, phase 1 studies, to identify, what was the minimum dose that would confuse patients. So it turned out to be 8 milligrams a day. What we found when we analyzed the plasma levels of patients in the trials, and this comes from over 1,000 patients, you can see along here the mean steady state plasma level of the drug in nanograms per mil. And here you have the decline on cognitive function over 65 weeks as measured by change in the ADAS-Cog score. So if you had minimal exposure at about the threshold was actually 0. The lower limit was just under 0.3 nanograms per mil. But the majority of patients receiving 8 milligrams a day showed they were in the range already needed for pharmacological activity. So that was 0.1. 0.2, pushing the dose to high levels didn't improve matters. So effectively, the trials were comparing 2 active doses which produced the same effect because like many other central nervous system drugs, there's a plateau. And, giving more antidepressant, giving more anti-epileptic, and so on, doesn't help more. It simply introduces more side effects. So the optimal dose from this study was 16 milligrams a day. And the other important thing that we learned from all of this data was that monotherapy, LMTM, hydromethylthionine, this is just the chemical shorthand for it, works much, much better than add-on therapy, about twice as well. We got exactly the same pattern of results when we looked at whole brain volume atrophy over 65 weeks. So at about 0.3 nanograms per mil, if that was the steady state plasma level, the decline was about 25 to 30 mils of brain substance, gray matter substance over 65 weeks. And again, you've got exactly the same profile of response. These two are highly correlated, the effect on brain atrophy and the effect on clinical decline. We've recently done a meta-analysis to examine, really to confirm the design assumptions of the ongoing phase 3 study. So here are 12 studies available in the public domain over 12 months. So it's almost 4,500 subjects from 12 published or available data sources. So this is the meta-analysis, and this is the weighted mean of all of that. And if we now look at low and high exposure at the 8 milligram a day dose, so this is what I showed you before, low and high exposure, using this threshold. This threshold was actually the lower calibration limit of the assay. We didn't invent this threshold. It's just the lowest limit of the assay detection. So if we look at, here is the meta-analysis summary, this value. So mild, moderate AD declines pretty well by just over 5 ADAS-Cog units over 12 months. So if patients had low exposure, so sub-threshold exposure, they declined exactly as expected. If they had high levels as add-on therapy, they did significantly better. And this is the p-value here is 0.300. And as monotherapy, much, much better. In fact, they didn't decline over 12 months. This is what the data shows. So here are the patients with low exposure, declining exactly as expected. Here is the drug as add-on. This, as I've shown you, this is a significant effect. And as monotherapy, there is no decline. We've compared this with the data available for aducanumab, but in this case, we had to look at mild AD. And so in mild AD, there are somewhat more studies, so 5,500 data from 5,500 patients. And interestingly, this is the placebo decline. So this is the meta-analysis again. The placebo decline in the aducanumab studies is more or less as expected. And here is the therapeutic dose, 10 milligrams a day. You can see it's well within the range of variability in expected placebo decline. And here is LMTM, hydromethylthionine, as add-on therapy or as monotherapy, clearly different. So we also looked at ADAS-Cog. Now we're going back to mild moderate. ADAS-Cog and activities of daily living. Here there's less data available, data from 1,300 patients from these studies. And you can see what the weighted mean placebo decline is. So if we now put those together, this is just repeating what I said before, that if you have patients with low exposure, decline as expected, but they also decline as expected on activities of daily living. As add-on therapy, it's significantly better, not quite significant for ADL, and as monotherapy, highly significant differences. Now, the question of ARIA has become very important because ARIA, amyloid-related imaging abnormalities, affect 35% of patients at the taking aducanumab at 10 milligrams per month. We actually looked at this in great detail. It was, in fact, one of our treatment emergent adverse events of special interest. And here you are. This comes from data from patients where MRI scans were conducted every 3 months over 15 to 18 months. So this is the composite data from the two studies. So you can see that the probability of ARIA at the 8 milligrams a day or at high doses, 150 to 250 milligrams a day, is 0.6%. This is within the range of placebo decline. So if we look back at past placebo data, Doody et al., 0.4%, edema, ARIA-E, and 5.6% microhemorrhages, and in the bapineuzumab study, 1.1%. So we are well within the range of expected placebo decline. So in short, the drug does not produce ARIA. Other parameters that were of interest to us, for example, serotonin syndrome, methemoglobinemia, very, very low incidence. So the study that's now ongoing is actually the only phase 3 tau-based trial currently ongoing. There are a few tau-based trials at phase 2. These are they're indirectly tau-based. This one's directly tau-based. The light blues are what are called proteostatic treatments, but everything else is built around, at phase 3, certainly amyloid, and the predominant phase 2 studies are amyloid-based. So that's it. That's my presentation. It's summarized, actually. I've sent Professor Mimica the presentation, but there's this lovely poster that if you contact the company, it's very well done. And a lot of the illustrations I've shown you are here. And if you ask nicely, our public relations people, they'll send you one of these. And in any case, you can get it online at targetingtau.com. So that's it. So thank you for your attention. And let me just, I should wrap up by saying, so the immediate prospect is if this phase 3 trial confirms what we already know from the completed phase 3 trials, then what will be available in the near term is an oral, ARIA-free, disease-modifying treatment that has the potential to produce a large treatment effect. So this will have a transformative impact. The cost of it will, I don't yet know what the cost is, but it certainly won't be $56,000 per patient year that Biogen want for their product. And payer organizations are not willing to reimburse largely. So, because the treatment effects are trivial. So this is very near now, and we hope sincerely that we can finally get this across the line for the benefit of patients. Okay, so that's my talk. Please feel free to ask questions.